The sieve tubes and the companion cells put together are called as sieve elements, which are the conducting elements of phloem in conducting food from upwards to downwards. Phloem is a food conducting tissue which transports or conducts foods from upwards to downwards, not from downwards to upwards. Potassium ions play a very great role in the opening and closing of stomata. So the stomatal opening and closing is a regulated process, controlled process. Hello everyone. Moving on to the last concepts of this interesting chapter called transport in plants. Moving on to the session 6 of this chapter 11 called transport in plants. Myself, B. Shobharani from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. Moving on to the remaining concepts of this very interesting chapter called transport in plants. As I said, the knowledge what we gain from the learning of these concepts is really admirable, because there are a good number of plants that are existing on this planet Earth, and we do not know what is the mechanism that is involved in the transport of water and dissolved mineral solutes, which we see in everyday life. But we do not try to understand the mechanism that is involved in the transport in plants. So all the concepts what we have learned in this particular chapter is definitely going to give us knowledge about or enlighten us about how the transport of the water and dissolved mineral solids in plants is taking place. So now let us, I think in the last session, we were discussing about a very important hypothesis called mass flow hypothesis, or it was also called as Munch flow hypothesis, because it was put across by Ernest Munch in 1930, where we came across the leaf as a source and the root as the consumption end or the sink. So what is the summary of this pressure flow hypothesis or mass flow hypothesis? As I said, it can be called as mass flow hypothesis or pressure flow hypothesis or Munch hypothesis. Hypothesis means what? It is just an assumption, like it might be due to. So it is just an assumption. So let us summarize the concept of this pressure flow hypothesis. So what is this? The summary is like this. The movement of sugars in the phloem, as we know, phloem is a food conducting tissue, and leaves, and particularly the mesophyll area of the leaf which prepares the food, prepared food is called starch, which is a polysaccharide, is carried by phloem, and that is carried by its phloem elements, namely the sieve cells and the sieve tubes, along with the companion cells. As we know, the sieve cells, the sieve tubes and the companion cells put together are called as sieve elements, which are the conducting elements of phloem in conducting food from upwards to downwards. So the movement of sugars in the phloem being at the source where sugars are loaded into the sieve tube. So the movement of sugars in the phloem being at the source. So source is the leaf where the food is prepared. So from there, the sugar, as I said, a polysaccharide, starch, the prepared food of plant is called as what? Starch. Starch, which is a polysaccharide. Polysaccharide is a carbohydrate. And we use a word called sugar. As you know, starch is insoluble in water. So starch is converted into glucose. Then it is soluble in water. So sugar, we call it as what? Glucose. As we know, starch is not soluble in water, and the main conducting or the medium is water. So the solids get dissolved in water, as water is the medium which is in liquid form, carries from one end of the plant body to the other end. So the movement of sugars in the phloem being at the source where sugars are loaded into where? Into the sieve tube. Loading of the phloem sets up a water potential gradient that facilitates the mass movement in the phloem. So there is a what you call it as the water potential gradient that is created when the sugar is loaded into the sieve tubes. So what is vein loading? Vein means what? As we already studied in the previous chapter, vein is nothing but which carries the vascular tissues. Vascular tissues, namely xylem and phloem, namely xylem and phloem. So what is vein loading? So what is vein loading? Transport of assimilates. Assimilates means all the contents that are dissolved, chiefly sucrose solutes from photosynthetic cells of the leaf, that is the source, into the sieve tubes of phloem with the help of ATP is termed as vein loading, or it can also be called as phloem loading. This is a very important term from examination point of view. So it might be asked like what is vein loading or what is phloem loading. So what is vein loading? Transport of assimilates, chiefly sucrose solutes from photosynthetic cells of the leaf. As we know, leaf is called as the source, and the sugars that are prepared in the leaf are loaded into the veins, namely the phloem. As we know, phloem is a food conducting tissue. To the sieve tubes of phloem with the help of ATP is termed as vein loading, or it is also called as phloem loading. So what is the summary of pressure flow hypothesis and what is vein loading? The movement of sugars in the phloem begins at the source where sugars are loaded, that is actively, what do you mean by loading? Actively transported into the sieve tube, and loading of the phloem sets up a water potential gradient that facilitates the mass flow movement in the phloem. So what is vein loading? Transport of assimilates, chiefly sucrose solutes, from photosynthetic cells of the leaf into sieve tubes of phloem with the help of ATP is termed as vein loading, or it is also called as phloem loading. Then ringing or girdling experiment. This is a very, very important experiment which is done to say that phloem is a food conducting tissue. Phloem is a food conducting tissue. This is an experiment called girdling experiment which is done to prove that phloem is a food conducting tissue. Right. So here, so you can see this experiment. This experiment is called as ringing or girdling. Why it is called ringing? Because you can see this particular part of the bark. Part of the bark is removed, and it is in the ring form. So you can see the ring form like so, ring like so, ring. So this is called as a ring. So here. So hence the name ringing or girdling experiment. So what is this experiment all about? A ring of bark. So the ring of bark on the stem of a small tree is removed so as to remove the phloem tissue also. So xylem is retained but phloem is removed. So here phloem from this particular part of the bark of a small tree is removed. As what is our experiment? Our experiment is to prove that phloem is a food conducting tissue. So food is transported from phloem from one part of the plant body to the other plant, that is from the source to the sink. So a ring of bark on the stem of a small tree is removed so as to remove the phloem tissue. So this is the first step. But only the xylem remains intact in that region. So xylem is there. As we know, xylem is a water conducting tissue, but our aim is to prove that phloem is a food conducting tissue. So we have to remove only the phloem part from this particular part of the bark of a tree, retaining the xylem. Then this process of removing bark in the form of a ring is called girdling, because we remove that particular part of a bark, retaining xylem and removing only the phloem. Hence it is called as girdling experiment. After few, so keep the whole apparatus in the sunlight and allow the process of photosynthesis to take place. Only the whole plant should be intact. Only that particular part of the bark, the phloem tissue of the bark is removed. So keep the whole plant in the sunlight and allow the process of photosynthesis to take place. So obviously the leaves will manufacture the food, and the prepared food is called starch, which is converted into sugar, and that sugar flows down. But it flows up to here, phloem is present. Phloem is present, and this particular part, phloem is absent. So with this girdling experiment, only one part of the phloem is removed. The upper part of the phloem is retained. So from the source, that is from the leaf, the prepared food called sugar passes from the leaf up to here. From here or there on, downwards it cannot be conducted. Food cannot be conducted. Why? Because phloem is absent. So what will happen? All the food that flows down up to particular this region gets accumulated. So food gets accumulated. And here you can see it swells up. Why it is swelling up? Because there is no way for the phloem to go conduct downwards because we have removed phloem tissue. So with this very simple experiment, what is the ultimate result, aim of this experiment? To prove what? That phloem is a food conducting tissue. So after a few days, it is observed that the region of the tree trunk just above the girdle is swollen. So this particular part is swollen. Right, I repeat, what is this experiment all about? What is this experiment called? Ringing or girdling experiment. What is the aim of the experiment? To prove that phloem is a food conducting tissue. So what is the method done here? A small tree is taken and one part of the bark, from one part of the bark region, phloem tissue is removed. The upper part of the bark region is remained. Then the whole apparatus, because we remove only one part of the phloem tissue from that particular part, hence it is called ringing or girdling. Then keep the whole apparatus in the sunlight. So allow the process of photosynthesis to take place. So obviously the sugar that flows from the leaf will get accumulated in this particular part and get swollen up. Why it gets swollen up? Because there is no way for it to go downwards because we have removed phloem tissue. So all the sugar gets accumulated in this particular part where phloem is retained and it swells up, but it is not able to flow downwards. So which is a very clear indication that phloem is a food conducting tissue. Hence it is called as what? Ringing or girdling experiment. So this is due to what? Accumulation of sugar in this particular region. Such accumulation is due to translocation of solids from the leaves downwards the root system. As I said, phloem is a food conducting tissue which transports or conducts foods from upwards to downwards, not from downwards to upwards. So the food molecules could not be translocated below the girdle because the phloem tissue is rich. So here, why only food is retained here? Swollen and sugar is accumulated. Why it is not accumulated? Not accumulated. Because there is no space, because phloem is removed. Phloem is removed. So this particular part is swollen because why? Sugar is accumulated in that particular part, phloem is there. But there is no other phloem for it to conduct downwards. So because the phloem tissue is removed in this region, after some time the root below the soil without getting food and the entire plant also dies. Yes, see, food is the prime required, may it be to a plant body or to an animal body. So there is no supply of food from the source to the sink, which is the root, then obviously the plant is going to die. So with this simple experimental setup, see how nicely they have proved that phloem is a food conducting tissue. Now moving on to the differences between transpiration and guttation. So already we have studied about transpiration and guttation. What is transpiration? How do you differentiate transpiration from guttation? Let us see one by one. Already I have explained what is transpiration and what is guttation in detail. Now let us summarize the differences because this is one such question which is commonly asked in the examination. So this is a very repeated question which is asked. Which commonly appears in the examination from this particular chapter called Transport in Plants. So what is transpiration? Normally occurs during daytime. Here usually occurs in the night or early morning. Then water given out in the form of water vapor. Here water is given out in the form of liquid water droplets. Then it is called as transpired water. It is called as guttated water, which is impure. Here the transpired water is pure. Here the transpired water. What is transpired water? The water that is expelled out in the form of water vapor, we call it as transpired water, which is pure. Whereas guttated water is impure, that is it contains solutes. Then it occurs through stomata, lenticels and cuticle, as I said, but it occurs through specialized structures called what? Hydathodes. Then it is controlled phenomenon called stomatal action. So, it is like, the opening and closing of stomata is regulated by the influx and efflux of potassium ions. So, potassium ions play a very great role in the opening and closing of stomata. So, the stomatal opening and closing is a regulated process, controlled process. And it is an uncontrolled phenomenon. So, this is called as an uncontrolled phenomenon. So, now let us see one by one. What is the difference between transpiration and guttation? Transpiration normally occurs during daytime. usually occurs in the night or morning. As we know, early morning we see the water droplets on the tip of the leaf or on the margins of the leaf, because the structures called hydrothodes are found on the margins and the leaf tip. So it normally occurs during daytime, usually occurs in the night or early morning. Water is given out in the form of water vapor. Water is given out in the form of water droplets or liquid form. And the water that is expelled out through transpiration is called transpired water, which is pure. Whereas the water that is transpired, or the water that is comes out through guttation, is called as guttated water, which is impure because it contains solutes. So it occurs through stomata, lenticule or cuticle. As I said, the transpiration is of three types. Stomatal transpiration, if the transpiration takes place through stomata. Lenticular transpiration, if the transpiration takes place through lenticels. What are lenticels? Ruptured epidermal cells. Or cuticle, which is the outermost layer, which is protective in function. Sometimes, as I said, maximum amount 98% or 99% takes place through stomata, and 1% through cuticle, and 0.5% through 1% through lenticels, and 0.5% through cuticle. So it occurs through hydrothodes, structures called hydrothodes. It is a controlled phenomenon because, as I said, the opening and closing of stomata is a regulated process, which is regulated through the entry and exit of potassium ions. So potassium ions play a very great role, as we have already studied in a chapter called mineral nutrition, that how important the mineral nutrients are for the growth and for the growth and development of a plant body. So it is an uncontrolled phenomenon. Hope you have understood all the concepts what I have explained in this very interesting chapter called Transport in Plants. So with this session, I have completed a very interesting chapter called Transport in Plants. So I'll be back in the coming session with yet another interesting chapter. Till then, goodbye and thank you.